Hi there, welcome to AP Biology. This is Anne Marie Hannibal. I am going to talk today about water and carbon. So I'm going to share my screen. So we want to share this one. Let's see. Okay, so let's take a look at water and carbon. Last class, we did a properties of water lab. And so we're going to talk a little bit about that before we get into the properties of water. What we noticed last time was that it looked like we could fit more drops of water, distilled water, on a penny than we could soapy water. So when we graph that data, it also looks like we can fit more drops of distilled water than we can distilled water with soap. So, and we can also see that when we graph our error bars, we're also getting no overlap between our error bars. And the most important thing about this lab to understand is that when error bars do overlap, you cannot say that there's a difference between your two data sets. But in this case, we do not have the error bars overlapping. So we can make a claim saying that distilled water holds more drops of, distilled water can hold more drops of water on a penny than the distilled water with soap. But if our error bars had been overlapping, then we could not make that statement. But we can because the error bars do not overlap. So our claim would be you can fit more drops of distilled water on a penny than you can distilled water with soap. And our evidence would be our averages or our mean, and then our SEMs, so plus or minus 2 SEMs, would also be our evidence. And then our reasoning would be an explanation of how the means are different and the error bars do not overlap. And because we are seeing more drops of water on a penny with the distilled water, we can say that there is stronger hydrogen bonding between those two, and we're seeing more cohesion and adhesion with the drops of water on a penny. But the soap seems to be disrupting those hydrogen bonds, and we're no longer able to hold as many drops of water on that penny. Okay, and then I've collected your labs. All right, so let's take a look at water and carbon. So we talked last class about water being the biological medium on Earth. So you kind of want to think about, what does that statement mean? What does it mean that water is a biological medium on Earth? What it means is the molecule can support life. It is the medium for all of those chemical reactions to take place on the planet. We need water so that those molecules can move around and they can allow those chemical reactions to take place. And so that's why most cells are made up of mostly water. They're also surrounded by a lot of water. And so all living organisms require that molecule more than any other substance in order to sustain life. So when they're looking for life on other planets, they're going to be looking for water to see if there's water present. If there's water present, then it is possible for life to exist there. So the biological medium of life on Earth is water because it facilitates all of those chemical reactions necessary for life. And that all is due to the fact that the water molecule is a polar molecule. And water, as you know, is made up of one oxygen and two hydrogen bonds covalently bonded, one oxygen, two hydrogen atoms covalently bonded together. And the reason why it's polar is because oxygen is more electronegative than the hydrogens, and it keeps those electrons closer to the oxygen side of the molecule. And then that leaves the hydrogen side slightly positively charged. And that is what a polar molecule is. So the little symbols at the top, this means this little squiggly symbol, it means partial. So the oxygen end is going to be partially negatively charged, and the hydrogen ends are going to be partially positively charged. And any polar molecule is going to have that charge difference between the sides of the molecule. And so water is a polar molecule, and that's what gives it its ability to hydrogen bond to other atoms. So when you've got two water molecules facing each other, the partial positive end is going to be attracted to the partial negative end. And that is what forms that attraction is called a hydrogen bond. So that attraction between the partial positive and the partial negative is the hydrogen bonding. And the hydrogen bonds allow those water molecules to stick to each other. And so that's cohesion of the water molecules. So each water molecule can be attracted to another water molecule with those hydrogen bonds. And that polarity of the water molecule is what gives it its unique properties. And there are 4 emergent properties of water that contribute to the Earth's sustainability for life. And those 4 properties are cohesive behavior, the ability to moderate temperature, expansion upon freezing, and versatility as a solvent. So if we look at each of those, we're going to first look at that cohesiveness of water. And cohesion is when water molecules stick to each other or hydrogen bond to each other. And that is what allows water molecules to travel up a straw. That's what allows water molecules to travel up a tree. So for trees and plants, they're going to be absorbing the water through their roots, and it's going to travel up the plant, and then it's going to evaporate through the leaves. And that allows for that flow of water through the tree. And it's the cohesion of the water molecules that allows them to stick together. And then adhesion is when the water molecule is attracted to another molecule, another substance. So the polar nature of the water is going to allow it to hydrogen bond to other polar molecules. So any molecule that is polar will have that attraction with the water. And then that again allows the water to travel up the tree. So it's going to stick, the water molecules are going to stick to each other, cohesion, but it's also going to stick to the sides of the tree as it transplants or transports the water up the xylem of the tree. Cohesion is also at the surface of the water, and that is referred to as surface tension. So surface tension is cohesion of the water at the surface, and that's what allows the water molecules to form into water droplets and allows animals like the water strider here to walk on water. So surface tension is cohesion at the surface, and it makes that surface of the water hard to break. So if you see in this picture here, all of these water molecules at the surface, they're all hydrogen bonded to each other. And then of course they're hydrogen bonded to each other within that droplet as well. So that's kind of what it would look like as we're putting those drops of water on a penny. Capillary action is, again, cohesion and adhesion of the water as it is traveling up a thin tube. Like xylem in the tree, but if you just had a very thin tube, the water is going to climb up it because of cohesion and adhesion. The thinner the tube, the higher up the water will travel through the capillary action. And so adhesion and cohesion, capillary action is what allows water to be sucked up through a straw. So to sum that up real quick, cohesion, water molecules sticking to other water molecules to each other, and adhesion, water molecules sticking to other molecules. All right, the second property of water that we're going to look at is the high specific heat of waters, which allows the moderation of temperature by water. It allows aquatic ecosystems to regulate their temperature, but it also allows organisms to regulate their body temperature as well. So specific heat is specific heat of a substance is the amount of heat that can be absorbed or lost for 1 gram of that substance to change by 1 degree Celsius. So to move the temperature of water by 1 degree Celsius for each gram, that's pretty high for water. So water has a very high specific heat. And that means that it is very resistant to changing temperatures. So it's very difficult to change the temperature of the water significantly. And that allows aquatic ecosystems, oceans, lakes, streams, rivers to maintain a relatively stable temperature. It doesn't change the temperature very much, which allows aquatic organisms to survive in the water because it is relatively stable. And we're also doing that for our body temperature. It helps us to regulate our body temperatures because it takes a lot of energy to change the temperature of our bodies, the water. And then also, when you think about when we are hot, think about what we do when we're hot. Also, due to high specific heat, evaporative cooling takes advantage of that high specific heat. So it really also takes a lot of energy to change water from a liquid state to a gaseous state. So that heat of vaporization allows our bodies to use that to cool our bodies. So evaporative cooling is taking advantage of that high heat of vaporization. So as we sweat, the water evaporates off of our skin and it uses our body heat to do that evaporating. So we sweat, our body heat is used to evaporate the water, and then it cools our body down. So it helps us to keep a stable body temperature. And that's all because of the high specific heat of water. And then the third property of water that we're going to take a look at is ice. And if you think about it, think about why is it important for ice to float? Think about living things. Why is that important? It's not really so important in our iced tea, but it is important to living things. So if ice didn't float, like a lot of other substances, when it changed from a liquid to a solid, it would be more dense and it would sink. If that were the case for ice, as the winter came and it started to freeze, the ice would sink and it would crush all of the organisms that were in the lakes and the streams and the rivers. We don't want that. We need those organisms to be able to survive in that aquatic ecosystem. And so it's important for the ice to float to allow aquatic ecosystems to survive the winter. So how that's possible is, again, hydrogen bonding. When water cools, so as you know, when water molecules are hot, they're moving around very, very fast. And those molecules are bouncing off of each other and they're spread apart. As the water cools, those water molecules will get closer and closer and closer to each other. And they get really, really close together at 4 degrees Celsius. So water is its most dense at 4 degrees Celsius. And then what happens is it starts to freeze and it expands, and those hydrogen bonds lock into a lattice type structure. So if we look at this picture here on the right, the liquid water is at 4 degrees Celsius. These water molecules are very, very close together. And then as it starts to freeze over here on the left, you can see how they're spread out more in this locked in lattice structure. And because they're in that lattice structure, there's space between those water molecules, and that is making it less dense. So this on the left, the ice is going to float because on the right here, those water molecules are more close together, and so they are dense. And so ice floats on the 4 degrees Celsius water, and it allows organisms to survive the winter underneath them. And then property number 4 is that it is a universal solvent. Water is a solvent of life. It is considered the universal solvent because it dissolves a lot of substances. It does not dissolve everything, but it will dissolve anything that is polar or charged. And so it's a very versatile solvent. And even if you are looking at substances that have an ionic charge, like if we look at sodium chloride, table salt, that is made up of sodium and chlorine ions that have crystallized. When we dissolve it in water, what happens is the chlorine, which is negatively charged, each and every chlorine ion is going to get surrounded by the hydrogen of the water that is partially positively charged. So the partial positive charge of the hydrogens is going to be attracted to the negative chlorine. The sodium is going to be, because it's positively charged, surrounded by the negative side of the water molecule. And so each and every one of those ions will get surrounded by the water molecule, and that is called a hydration shell. And then that allows every single one of those ions to dissolve in the water. And that's going to be the case for other molecules as well. So if you're talking about sugar, protein, large polar molecules, those can also get dissolved in water if there are enough polar regions for the water to surround it and dissolve it. So sugar is going to dissolve in water. The proteins are going to dissolve in water. Any molecule that is polar is going to get dissolved in that water because the water will form a hydration shell around those molecules. If we look at nonpolar things like oil, as you know, oil and water don't mix because oil is nonpolar and water is polar. They're not going to mix together. So hydrophilic substances are ones that have the affinity for water. They can dissolve in water. They will dissolve in water, but hydrophobic substances are not going to have that affinity for water. They are not going to dissolve in water because they are non-polar molecules. So water dissolves a lot of things, but it does not dissolve everything. Okay. And if you think about the cell is made up of mostly water, both inside and outside of the cell, and that allows, again, those chemical reactions to take place in the water. So those molecules are moving around in the water, and those chemical reactions depend on that movement, that motion in the water, and therefore allows for those chemical reactions. So we need that aqueous solution inside the cell, outside the cell, so that those chemical reactions can take place. And then just a real quick discussion of acids and bases, because again, we're talking about dissolving water, substances in water. And water goes from a dynamic equilibrium, the state of water molecules, into these ionic forms. So a hydronium ion is going to have an extra hydrogen, and a hydroxide ion is going to be missing a hydrogen. And it's kind of in dynamic equilibrium. It goes back and forth. But if you add a substance to water that increases the hydronium ions or the hydrogen ions in that water, that is an acid. And if you add something to water that increases your hydroxide ions, then that would be considered a base. So acids increase the hydrogen ion concentration of the substance, and bases increase the hydroxide ion substances in that water. So that's really all we're going to talk about with acids and bases for now. We're going to leave most of the acids and bases to chemistry, but we'll see hydrogen ions and hydroxide ions throughout the year. Okay, so that is water. And then we're going to talk a little bit about carbon today, and then we will leave the macromolecules for another lecture. So if we look at carbon, carbon is considered the backbone of life. And you kind of want to think about what properties of carbon are there that makes it that backbone of life. All living organisms are made up of mostly carbon. It is a large and complex molecule, or it can make large and complex molecules. So proteins, DNA, carbohydrates, and lipids, all of those macromolecules of life are composed of carbon compounds, carbon bonded together in all kinds of different combinations. So if we look at why that is the case, we're kind of looking at organic chemistry. Organic chemistry is the study of carbon compounds. So anytime you're combining a whole bunch of carbons together, that is organic chemistry. And you're looking at the interactions and the reactions with those carbon molecules. So what makes it so unique is its ability to make those large complex molecules. So if we look at hydrogen, hydrogen has the ability to make one covalent bond. Oxygen has the ability to make two covalent bonds. Nitrogen has the ability to make three covalent bonds, but carbon has the ability to make four covalent bonds. And that is what makes carbon so special. It can make four covalent bonds. And because it can make four covalent bonds, you can bond it together in all kinds of different arrangements, and you can make short chains, you can make long chains, you can make branched chains, you can make ring structures. And that ability to make those chains in different lengths and those rings and combine rings together, all of that is what makes carbon so special, because you can arrange it in so many different ways. And that is what allows that foundation for all of those macromolecules. Now, hydrocarbon is simply an organic molecule that is made up of mostly hydrogens and carbons. Hydrocarbons tend to be nonpolar, and so therefore not dissolving in water. And we'll talk more about hydrocarbons when we get to macromolecules. But a hydrocarbon is really an organic molecule consisting of primarily carbon and hydrogen. And then real quick, we're going to look at isomers. Isomers are just different arrangements of molecules. So they're going to have the same molecular formula, but they're going to have a different structure. And because their structure is different, it gives them a different function or properties. And so in biology, structure and function go hand in hand. It's very, very important to understand that the structure of the molecule is related to how it works, how it functions in the cell. And so different structural arrangements are going to have different properties, and they're going to work differently. And so a structural isomer is simply one that has different covalent arrangements of the atoms. They've got the same number of atoms, they're just arranged differently. And when we look at cis-trans isomers, you're talking about double bonds between those carbons, which are going to make where you put those functional groups, they're going to make them function differently. So if they're on the same side of the double bond or opposite sides of the double bond, they're going to work differently. So it does matter when you have double bonds where you're attaching things to those molecules. And so those are cis-trans isomers. They're going to have the same covalent bonds, but they're going to be arranged differently in a spatial arrangement. And then enantiomers, again, they've got the same molecular formula, but they are arranged differently. So if you look at your left hand and your right hand, they look very much the same, but they function differently because they are mirror images of each other. So if you are right-handed, it's very difficult to write with your left hand, and vice versa, because your left hand and your right hand they're not exactly the same. So they function differently, even though they look the same. And so those different isomers are just going to have different functions. And then there are functional groups that we can add on to these biological molecules. And functional groups are just groups of atoms that will have certain functions, and they're going to give each molecule a function based on the properties of that particular functional group. You don't have to memorize those functional groups, but you do need to get familiar with them. And we will get familiar with them as we are exposed to different functional groups throughout the year. So as we go through the different macromolecules and the different molecules throughout the year, we will see different functional groups and how they affect the different functions. So here, just for example, if we look at these two are steroids. They look very similar, but they've got different functional groups attached onto them. So they're going to work very differently. They're both steroids, but they're going to have a different role in the body because of the different functional groups that they have attached onto them. So there are 7 functional groups that are important when we're looking at biological molecules. But again, you don't have to memorize them. We will see them as we go through the year. But the hydroxyl group is an OH group, so we're definitely going to see that attached onto molecules. And when we talk about dehydration synthesis next class, we will see those hydroxyl groups come into play. A carbonyl group is going to be a carbon with a double bond to an oxygen. We've got that was a carbonyl. A carboxyl, we will see when we talk about amino acids and bonding those together and the functional groups on that. An amino group is a nitrogen with 2 hydrogens. Again, when we talk about proteins and amino acids, we'll see that functional group. Sulfhydryl is a sulfur and a hydrogen. We'll see that when we, again, look at proteins and our group interactions. Phosphate group, we will see when we talk about phospholipids and when we talk about DNA and ATP. So we'll see that one a lot throughout the year. Methyl groups, we'll talk about methylation when we get to our DNA chapter and gene regulation. So there are those functional groups, and the key to understanding is that those functional groups are important to understand the function of those macromolecules. So we will see those functional groups, and remember, in biology, structure equals function. The structure of those molecules is related to the functionality of those molecules. So very important to understand the shape is related to how it works. Okay, we are going to stop there and we will talk about macromolecules in another session. So I hope this was helpful and we will stop here.